Welcome to EPG Pathshala. I am Dr. Vinita Sondi from Vivekanand College. We will be discussing organic dysfunctions and crime. This module introduces the linkages between organic dysfunctions and crime. In the past 20 years, the field has grown tremendously and the influence of biological factors such as genetics, hormones, neurotransmitters, brain disease and diet on criminal behavior has been examined. This growth in part is due to the many new brain imaging techniques that have been developed. The module will introduce classical as well as new research in the area. However, it needs to be remembered that the area is still developing and many questions will arise with respect to the studies being quoted. The complex interactions between biological influences and other factors such as the environment makes it a challenging task to pinpoint clear-cut directions. There are certain learning outcomes that are expected out of the module. Upon completion of the modules, students will be able to describe the links between organic factors and criminal behavior, critically evaluate the genetic factors leading to criminal behaviors, understand the interplay of biochemical factors such as hormones and neurotransmitters with criminal behaviors, discuss how various brain diseases can affect aggression and violence, understand how dietary habits and toxins in food can lead to aggressive tendencies. Next, we will discuss the role of the various biological factors in criminal behavior. The first factor that we will discuss is genetic factor. The idea of an aggression gene or a criminal gene that is inherited has fascinated many researchers and spurred a lot of research in the area. Though researchers have not been able to identify such specific genes for criminal behaviors as yet, genetic predispositions to criminal behavior has had some support in literature. One method for identifying whether behavior has a genetic basis is through twin studies. These studies are based on the premise that monozygotic twins or identical twins share 100% of the genetic makeup, while dizygotic twins share only 50% of it. Hence, if concordance rates are higher for monozygotic twins, then this would indicate a genetic basis for that behavior. Concordance rates can be thought of in terms of percentages which show how many pairs share that behavior. Rain in 1993 analyzed some of the early twin studies and found a greater concordance rates for criminal behavior in monozygotic as opposed to dizygotic twins. One of the most famous sets of twin studies was conducted in Denmark. Denmark is known to keep official records of many parameters, including criminal activity. Christensen in 1977 examined over 3,500 twin pairs in Denmark and identified concordant rates of criminal behavior. The study found a higher concordance rate for monozygotic twins for criminal behavior than in dizygotic twins. This finding found some support for the genetic basis of criminal behavior. Other studies too have found results in the same direction. However, the results of such studies need to be treated with some caution. The twin studies are based on the equal environment hypothesis, which states that monozygotic and dizygotic twins share the environments to approximately the same extent. However, some researchers point out that monozygotic twins may be treated more similarly than the dizygotic twins, and this violates the equal environment hypothesis. One way of removing this problem is through adoption studies. Crow 1972 compared two groups of adopted children to understand the genetic basis of criminal behavior. One group of adopted children had biological mothers with a criminal record. Another group of adopted children acted as the control group and had biological mothers with no such criminal records. The study indicated there was a very high chance that children whose biological mother had a criminal record would also develop some kind of criminal record by the time they were 18, while for children whose biological mothers did not have any such past records, this chance was very low. This suggests that irrespective of the environment, there seemed to be a genetic predisposition to criminal behavior. One of the largest adoption study was carried out by Mednick et al. In 1984 in Denmark. They reported a significant correlation between adopted children and the biological parents with respect to criminal behavior. However, this correlation was found significant for property crimes but not for violent crimes. Another country where a number of such studies have been carried out is Sweden. Bowman and his colleagues 1978 carried out the Stockholm adoption study and found that criminal behavior alone of biological parents did not increase the risk of criminality in adopted children. However, there was some evidence for high rates of criminal behavior in adopted away offspring of biological parents with alcohol use disorders alone or with both alcohol use disorders and criminality. Bowman 1978. This seemed to indicate that crime and genetics were not directly linked but linked indirectly through alcoholism. One of the genes that has been identified vis-a-vis its role in criminal behavior is monoamine oxidase A, often referred to simply as MAO. This gene regulates an enzyme, monoamine oxidase, that degrades excess neurotransmitters like serotonin, dopamine and norepinephrine. However, one variation of the gene produces an inactive enzyme which is not effective in degrading the excess neurotransmitters. Moffitt 2005 found that children with this variant of the MAO gene had a higher risk of growing up to be antisocial or violent only if they were also abused as children. The expression of the violence gene changed if the family was loving and non-abusive, and such children did not grow up to be antisocial. Another gene, CDH13, is also linked with violent behaviors and with many clinical disorders such as ADHD, autism, schizophrenia and bipolar disorder. It has been suggested that changes in the expression of these specific genes in the brain such as MAO and CDH13 can affect neurotransmitter levels, which in turn influences behaviors such as criminality. However, environmental factors may interact with genetic factors in yet unknown ways. Thus, most of the studies seem to indicate a link between criminal behavior and genes, though others have questioned these findings. There are a number of questions regarding the studies such as violation of the equal environment hypothesis and sample size of the studies. Also, no single gene has been identified with criminal behavior. Instead, numerous genes are involved and interact with the environment in complex ways. The second factor which we will examine are hormonal factors. Hormonal imbalances can result in changes that range from minor mood swings to even death. Classical research in the area has been conducted by Dabbs and his colleagues in the 90s. In one study, it was found that men who committed crimes involving sex and violence had significantly higher testosterone levels than men who had committed property crimes. In another study, Dabbs and Morris investigated the relationship between testosterone levels, socioeconomic status and antisocial behavior. They searched records of more than 4,000 US military veterans and categorized them in two groups, higher socioeconomic status and lower socioeconomic status. These two groups were then compared in terms of aggression and antisocial behaviors. The study found higher testosterone was associated with delinquent behaviors in men of low socioeconomic status, but not in men of higher socioeconomic status. According to the researchers, higher socioeconomic status individuals may be able to express their dominance in many different ways rather than simply aggression. Therefore, the chances are much lower that they will be in criminal trouble. In a prison study of female prisoners, Dabbs et al. Found that testosterone levels and violence in women were closely linked with testosterone levels, with levels being the highest in female prisoners who had committed unprovoked assaults and lowest in female prisoners who had only reacted violently when they were physically assaulted. Van Goosen et al. 1997 studied people undergoing sex change operations. The study indicated that women undergoing a sex operation to change into men and who got testosterone injections became more aggressive and sexual. On the other hand, men undergoing a sex operation to change into women who got testosterone suppressants became less aggressive and less sexual. Although there is quite a lot of evidence which indicates that testosterone levels are closely related to aggression, violence and antisocial tendencies, but questions have been raised regarding some research findings. Not all studies have found similar findings. Olvius et al. 1988 conducted a comparative study of institutionalized delinquent boys and non-delinquent male students. The study indicated no statistically significant differences in levels of testosterone between the two groups. However, they observed that those adolescents who had higher levels of testosterone had higher chances of taking up challenges. Much of this evidence is based on correlational data, and correlation does not mean causation. High hormonal levels can lead to high levels of aggression, but high levels of aggression can also lead to high levels of the hormone. Moreover, the importance of environmental factors in causing aggression has been overlooked in many of the studies. The links between testosterone and aggression have been found in males, but many studies indicate that the results cannot be generalized to females. Researchers have additionally pointed that testosterone can also have a positive influence. For example, McNicholas et al. 2003 found that testosterone replacement therapy increased positive mood and decreased negative mood. These findings have been supported by Zitzman and Nishlak 2006, who also found that testosterone supplements in older males produce positive results and increase vigor and energy. Another hormone that has been implicated in aggression is cortisol, but the role of cortisol is more complicated than that of testosterone. Many researchers like King et al. 1990 have suggested that there is an inverse relationship between the two, with low levels of cortisol associated with high levels of aggression. It has been hypothesized by Rain 1996 that people who have low levels of cortisol have lower autonomic arousal. In order to increase the arousal of the autonomic system, these individuals then seek stimulation which is inherent in stressful situations. Stressful situations in turn result in autonomic nervous system activation and cause cortisol to be released. However, the research regarding the link between aggression and cortisol levels is not clear cut. Some studies such as those by McBurnett et al. 2000 have found support for an inverse relationship between cortisol and aggression, while others such as those by Guerra et al. 1997 have found a direct relationship between the two. The third factor that is important in understanding criminal behaviors is neurotransmitters. The role of serotonin and dopamine has been examined in the context of aggression. Many, though not all studies, indicate low levels of serotonin and high levels of dopamine are linked with aggression. The serotonin deficiency hypothesis of human aggression states that a deficiency of serotonin causes aggression, especially impulsive aggression. Impulsive aggressive behaviors include self-mutilation, suicide attempts, domestic violence and substance abuse. In one study, Brown et al. 1982 found a link between serotonin levels and aggression, with low levels of serotonin associated with high levels of aggression. In another experiment by Mann et al. 1990, administered dexfenfluramine to participants, which led to reduction in levels of serotonin in the brain. They then responded to a questionnaire that assessed aggression and hostility. The results indicated that males obtained higher scores on hostility and aggression on the questionnaire after taking the drug. This was not found to be true for females. The relationship between dopamine and aggression is more complicated. According to Kupis et al. 2008, some individuals have a dopamine system that rewards their aggression behavior, and as a result, they intentionally seek out aggressive encounters. However, dopamine interacts with other neurotransmitters in complex manners and controls many behaviors. Many of the studies have carried out in animals and hence cannot be directly extrapolated to human beings. Brain damage is the fourth important factor that has close links with aggression, violence and other criminal behaviors. A number of researchers have examined the brains of violent criminals using techniques such as MRIs, PET scans, fMRIs, etc., and have found a close link between violent acts and head injuries. Blake et al. 1995 observed that many of the violent criminals had suffered from brain injury, and about 64.5% of the accused murderers had some damage to the frontal lobe. However, studies like these are difficult to interpret because of the small sample size, absence of controls and other methodological factors. One area of the brain that has been examined with respect to criminal behavior is the prefrontal cortex. The prefrontal cortex exerts control over primitive reactions to environmental provocation. In other words, when something makes us emotional, areas in the prefrontal cortex become active, and this inhibits a violent or an aggressive response. It also helps us to see long-term consequences of our actions so that an impulsive response is inhibited. Frontal lobe damage can occur in a number of different ways, such as a car accident, sports, development malformation, alcoholism, tumors, strokes, etc. The role of the prefrontal cortex was examined in the Vietnam head injury study by Grafman et al. In 1996. The results indicated that veterans with temporal injuries reported more feelings of rage and hostility, whereas veterans with injuries to the prefrontal cortex, particularly the orbitofrontal cortex, reported a much higher level of violent and aggressive behavior. It needs to be mentioned that not all head injuries lead to violence and crime. It may be possible that head injuries result in increased aggression in those who are already predisposed to it due to other factors such as social and environmental. Thus, aggression and criminal behavior is a result of complex interactions between nature and nurture. Also, even in the unfortunate case of a head injury, various interventions exist which can reduce the effects to some extent as long as treatment starts early. The last factor that we will examine would be dietary habits and toxins. Normal human brain functioning requires higher levels of glucose per minute. Reduction in glucose levels can result in symptoms such as irritability, panic and aggression. There have been many studies like those by Rain 1993 that linked low blood sugar or hypoglycemia to aggressive behaviors. Depending on eating habits, many people appear to have a dip in their blood sugar around 11 to 11:30 am. And in prisons, this time corresponds with the majority of cases of inmate violence. However, these studies do not prove conclusively that a person was hypoglycemic during the commission of a crime. It is not possible to obtain glucose levels at the time of the criminal act, and hence not possible to prove this hypothesis. Moreover, the effects of low blood sugar vary in individuals. Some show no apparent effect, some feel dizzy, and some may panic. In addition, the effects of low blood sugar interact with a number of other factors that have been causally related to violence and crime, such as alcohol usage, serotonin levels, EEG, and low heart rates. It was stated earlier that low serotonin levels are linked to aggression. Serotonin is made from the amino acid tryptophan, which is derived solely from our diet. Thus, a diet low in tryptophan will result in low serotonin, while an increase in dietary tryptophan could reverse the low serotonin levels. Many experimental studies use the method of acute tryptophan depletion, which involves reducing the availability of tryptophan by administering a drink to understand the link between serotonin and aggression. Clear and Bourne, 1995, used the method of acute tryptophan depletion to understand the role of serotonin in aggression. Before taking part in the experimental procedure, the participants were assessed for their aggression. Two groups with high and low aggressive tendencies were differentiated. It was observed that those participants who had high aggressive tendencies prior to the experiment became more so after tryptophan depletion. However, tryptophan depletion had no effect on those having low aggressive tendencies. In another study by Moeller et al. 1996, a tryptophan-free amino acid mixture was given to healthy males to induce tryptophan depletion. The researchers observed a significant decrease in aggression due to the low tryptophan diet. However, the results point in a particular direction, but they are not conclusive as many unsolved questions still remain. Toxins in the environment have become very common and are even sometimes present in our foods. Many of these toxins are heavy metals such as lead, calcium, and manganese. The role of lead on aggression has been investigated by many researchers. Lead is used in many everyday products, including paint, makeup, bullets, fuel, and plumbing. High levels of lead in the body can be very deleterious to human health and can lead to brain damage as well. In one study, Ward 1995 successfully obtained hair and blood samples from a group of 68 young violent criminals who were males aged 16 to 19 years and compared them with a group of controls. Analysis indicated lead and aluminium levels in the young violent criminals. They were 4 times the levels in the control group, while the cadmium levels were doubled. The study also revealed deficits in certain essential elements including chromium, selenium, and zinc. Zinc and chromium were reduced by a factor of 4, which is the same factor as the toxic metals were raised. The ill-fated Franklin expedition is another important example of how high levels of lead could cause violent behaviors and lead to eventual death. Sir John Franklin's 1845 Arctic expedition began from England in the search for the Northwest Passage to Asia. The expedition had 2 very well equipped ships and 129 crewmen. The ships had loads of canned foods. In early expeditions, preservation of foods was not easy and many sailors would develop scurvy due to lack of vitamin C. However, both the ill-fated ships disappeared and no one was ever seen alive again. The local people spoke of the last days of the sailors and described acts of insanity, violence, and eventual cannibalism. Subsequent searches found frozen graves of 3 sailors on Beechey Island in the Canadian Arctic. Several tin cans were also found. Then in 1984, Dr. Owen Beatties, a Canadian anthropologist from the University of Alberta, exhumed the bodies from the grave. Beatty discovered lead concentrations up to 20 times more than normal in the bodies. The source of the lead was found to be poor technology used in canned food. The lead from the seal had leaked into the food. Some cans had simply exploded and sealed cans were heavily laden with toxic layers of lead. It is suggested by Beatty and Giger, 1998, that starvation and lead poisoning killed the men eventually. Feigenbaum and Mueller, 2016, observed that in the second half of the 19th century, lead pipes were used as part of the water systems in many American cities. What was the effect of lead pipes on homicide between 1921 and 1936? American cities having water pipes made of iron pipes had much lower homicide rates as compared to cities with lead pipes. They concluded that lead water pipes exposed entire city populations to much higher doses of lead, and this led to increased levels of homicides in American cities. In another study, Iser and Genetic&amp, 2017, observed that children living on busy roads were more likely to have high lead levels in their blood because of contaminated soil. This contamination is due to the use of leaded gasoline decades ago. They compared these children having high lead exposure with similar kids who lived on other roads. Iser and Curry concluded that exposure to high levels of lead increased the probability of suspension from school and of detention in boys. In light of these studies, many countries have rules and regulations that are aimed at reducing lead levels in the environment. Also, neurotoxicity is only one of the factors that is related to violence and aggression. Other risk factors include poverty, social stress, alcohol or drug abuse, even individual character, and maybe many more social factors. Let us now briefly summarize this module. A large number of biological influences on human behavior have been examined. Twin studies and adoption studies have found strong support for a genetic basis of criminal behavior. A few studies also seem to indicate that crime and genetics may not be directly linked, but linked indirectly through alcoholism. There are also a number of questions regarding the gene studies, such as violation of the equal environment hypothesis and sample size of the studies. Two genes that have been identified with links to criminal behavior are MAOA and CDH13. However, environmental influences can change the expression of genes. Some studies have found a link between criminal behaviors and hormones. Two hormones that have been researched include testosterone and cortisol, but the role of cortisol is much more complicated than that of testosterone. Some studies have found support for an inverse relation between cortisol and aggression, while others have found a direct relation between the two. Two neurotransmitters within the brain are implicated in aggressive behaviors. These are serotonin and dopamine. Many, though not all, studies indicate low levels of serotonin and high levels of dopamine are linked with aggression. Many studies have indicated a close link between violent acts and head injuries. One area of the brain that has been examined with respect to criminal behavior is the prefrontal cortex. There have been many studies that linked low blood sugar or hypoglycemia to violent or aggressive behavior. Other studies indicate that a diet low in tryptophan will result in low serotonin and consequent increase in aggression. Again, the results point in a particular direction but are not conclusive. The role of lead on aggression has also been investigated by many researchers. Studies indicate high lead and aluminium levels in young violent criminals. In conclusion, one can say that biological factors are linked to violence and aggression, but they interact with many other risk factors such as poverty, social stress, alcohol or drug abuse. Individual character and other social factors may also be important. Thank you.